Welcome to the Concierge Care Health System podcast. The information shared in this podcast is for general informational purposes only and does not constitute medical advice. Listeners should be aware the content is not a substitute for professional medical advice, diagnosis, or treatment. Always consult with a physician or other qualified healthcare provider for medical questions.
What if your brain simply forgot how to sleep? Not just, a rough night, but a complete, terrifying breakdown of the ability to ever sleep again. Today, we're plunging into an incredibly rare, devastating, and scientifically fascinating condition known as Fatal Familial Insomnia, or FFI. We've gathered some really comprehensive insights into this disease, covering everything from its molecular secrets right through to the profound human cost. Our mission today is to kind of cut through the complexity, connect the dots for you, and help you really grasp the essence of this unique neurodegenerative disorder, but without getting totally bogged down. Okay, let's unpack this.
Yeah, and one striking thing about FFI, it's so much more than just severe insomnia. It really is. It's a genetic prion disease. That puts it in a category of inherited neurodegenerative disorders that are, well, tragically, always fatal. Now, while it shares a family tree with other prion diseases like Creutzfeldt-Jakob disease, FFI really stands out. It stands out because it precisely targets a very critical brain region. And that targeting, in turn, offers these unique insights into the very mechanisms of sleep, consciousness, even how our basic bodily functions are controlled, The story of FFI itself is pretty remarkable, too, just cracking the medical discovery. You go from these mysterious familial curses in old reports like that 1765 account of an Italian man right through to its formal clinical description in 1986 by Lugarese. And then finally pinpointing its exact genetic basis. It's quite a chronicle.
That really is quite a journey, and it brings us right back to the start. What is a prion disease, fundamentally? And how does FI fit into that whole picture? I mean, we hear prion, maybe think mad cow disease, but how does that work for something like FFI?
That's a great question, and yeah, it's absolutely key. Prion diseases are caused by what's often called the protein-only hypothesis. Basically, there's a normal, perfectly healthy protein in our cells, especially abundant in the brain, called PRPC. Think of it like a normal working part in the brain's machinery. It's involved in things like cell communication. But in prion diseases, this normal protein gets corrupted, it misfolds, it changes shape into a pathogenic diseased version, which we call PRPSC. And here's the really insidious part. This misfolded PRPSC acts like a template. It forces other normal PRPC proteins it encounters to also misfold.
Wow, okay, so if I'm getting this right, it's like a normal protein goes rogue. And once it misfolds, it becomes this destructive agent that basically recruits others to do the same thing, spreading this sort of molecular chaos.
Exactly, you've got it. It's a chain reaction of corruption. And for FFI specifically, the root of this chaos lies within a particular gene, the PRNP gene, located on chromosome 20. The primary genetic defect for FFI is a tiny change, a single point mutation at a specific spot, codon 178. An amino acid called aspartic acid gets swapped for asparagine, a D to N switch. But here's where it gets really fascinating and where FFI teaches us so much about genetic precision. The outcome isn't just about that one single change.
You're talking about the molecular switch idea, right? How another common variation nearby changes everything.
Precisely. There's this common, completely normal genetic variation, a polymorphism at codon 129 in the same gene. Now, if that D178N mutation happens on the chromosome that also has the amino acid methionine at codon 129, you get FFI.
Yeah.
FFI is defined by this profound targeted degeneration of the thalamus.
Okay.
However, if that exact same D178N mutation is paired with a different amino acid, the thymine, at codon 129, then it results in a subtype of familial Creutzfeldt-Jakob disease, FCJD, which has very different symptoms, more widespread brain damage, especially in the cortex.
Wow.
It really shows how this subtle genetic context dictates not just if you get a prion disease, but which devastating form, and where it primarily attacks the brain. It's an amazing lesson in genetic precision.
That's truly wild. A single amino acid swap plus this common variation nearby completely redirects the disease. So, how rare are we talking here? Is this something many people carry?
No, not at all. FI is exceptionally rare. It's inherited in what's called an autosomal dominant pattern. That means each child of an affected parent has a straight 50% chance of inheriting the mutation.
Right, a coin flip.
Exactly. And we're talking about maybe only 50 to 70 known families worldwide who carry this specific mutation combination. Historically, there were clusters in Europe, Italy, Spain, Germany, and more recently, we're seeing a growing number of identified cases in China. It's also worth just quickly mentioning sporadic fatal insomnia, SFI. It looks almost identical to FFI clinically and pathologically, but it happens without that D178N mutation. The thinking there is it's likely caused by a spontaneous random misfolding of the normal prion protein, even rarer, usually.
Okay, so once this misfolding cascade kicks off, the specific prion strain is sort of set by the genetics. What's actually happening inside the brain? Why the thalamus? Why is that deep brain structure so specifically and uniquely vulnerable in FFI?
Right. So as that misfolded PRPSC spreads, it acts as that template, forcing more normal PRPC to convert. You get this exponential pileup of the bad proteins. These misfolded proteins clump together, they aggregate, forming structures from tiny soluble bits called oligomers up to larger amyloid fibrils, and these aggregates are profoundly toxic to neurons. They mess up essential cell functions like proteostasis of the cell's quality control system for proteins. This triggers stress responses inside the cell and ultimately leads to apoptosis or programmed cell death. And interestingly, many researchers now believe the smaller soluble PRPSC oligomers might actually be the most toxic species, not necessarily just the big visible plaques. The absolute hallmark of FFI, pathologically speaking, is that severe and incredibly selective damage to the thalamus. And the thalamus isn't just any brain region, it's like the brain's central relay station, or maybe a gatekeeper. It filters sensory info, regulates consciousness, sleep, alertness. It's critical. In FFI, you see massive neuronal loss, often over 80% in key parts of the thalamus, specifically the mediodorsal and anteroventral nuclei, and also parts of the brainstem. But here's the really weird part, what some call the pathological paradox. The thalamus is devastated, profound atrophy, but often it has surprisingly few visible PRPS deposits compared to other areas. You might find more visible protein clumps in, say, the cortex, even if it's less damaged overall.
That's fascinating and really counterintuitive. It suggests the visible stuff, the plaques we often hear about, might not be the main driver of cell death, at least not here. Maybe it's those smaller toxic forms, or just that thalamic neurons are uniquely susceptible for some reason we don't fully understand yet. That really shifts how you think about tackling it. So given that selective attack, can you walk us through the progression? What does this systematic dismantling actually look like for someone going through FFI? It sounds terrifying.
It is a truly relentless and frankly terrifying progression. Symptoms typically kick in around age 50, but the range is huge, could be 20s, could be 70s. Average survival is tragically short, about 18 months. But again, there's variations, 7 months up to maybe 73 months. And that seems to be influenced, at least partly, by that codon 129 variation we talked about, even on the non-mutated gene copy. People with 2 methionine copies tend to have a faster, tougher course.
Right.
The disease generally unfolds in 4 stages, more or less. Stage 1, maybe 3 to 6 months, starts kind of subtly. Worsening insomnia is key, but also growing panic attacks, paranoia, maybe weird specific phobias. Sleep gets fragmented.
Things you might initially brush off, maybe?
Exactly. You might think it's stress or something else. But then comes stage 2, lasting about 5 to 9 months. Psychiatric symptoms ramp up, vivid hallucinations become common. And crucially, you see this widespread autonomic hyperactivity. The body's automatic systems go completely haywire. Persistent rapid heart rate, high blood pressure, profuse sweating, high body temperature. Like the body's thermostat is just broken.
Wow, the body attacking itself almost.
In a way, yeah. The control systems fail. Then stage 3, usually around 3 months long. This is where you see total or near total insomnia. Sleep becomes practically impossible. Patients experience rapid, dramatic weight loss even if they're eating okay. And cognitive and motor skills decline sharply. Finally, stage 4 maybe lasts up to 6 months. This is the terminal stage. Severe dementia sets in. Patients often develop akinetic mutism. They can't move or speak. Eventually, coma, and then death.
It's, systematic dismantling.
It really is. It hits 3 core human functions: sleep, basic bodily control, and higher cognition. The main symptom, the name gives it away, is that profound sleep disturbance. It's called agrypnia excitata. It's not just can't sleep. It's a total breakdown of the sleep-wake cycle. Sleep studies show almost no deep sleep, no REM sleep. The normal brainwave patterns of sleep just disappear. Patients might get trapped in this one-eric stupor, acting out vivid, disturbing dreams while seemingly awake. And that's a direct result of the thalamus failing. It's the gatekeeper for sleep, and the gate is broken. Then there's the severe autonomic dysfunction, that fight-or-flight system stuck on overdrive, loss of internal balance. Motor problems also emerge: ataxia and unsteady walk, dysarthria, slurred speech, trouble swallowing, myoclonus, those sudden muscle jerks, sometimes Parkinson's-like stiffness. And underlying it all, the cognitive and psychiatric decline, problems with attention, short-term memory, planning. Mood changes like apathy, depression are common, plus that anxiety, paranoia, and those really complex hallucinations. It's a complete assault.
It's just an absolutely terrifying, almost orchestrated progression. Truly heartbreaking. So given how rare it is, and this complex mix of symptoms, how do doctors actually confirm it's FFI? What's the diagnostic path look like?
Yeah, you're right. Diagnosis definitely needs a high level of suspicion because it is so rare. It's really a multimodal approach, piecing together clues. The absolute gold standard, the only way to be 100% sure, is the genetic test. Testing the PRNP gene. You need to find that specific D178N mutation linked up with methionine at codon 129. That's the FFI signature.
Okay, so genetics is key.
Absolutely essential. But other tools are crucial for support and sometimes to raise suspicion in the first place. Polysomnography, the sleep study, gives objective proof of that profound sleep chaos. The loss of deep sleep, REM, the totally disorganized structure. Functional brain imaging, especially PET scans, is another big one. PET scans consistently show this characteristic pattern. Severe hypometabolism, meaning much lower energy use, specifically in both sides of the thalamus.
The thalamus shutting down.
Exactly. It's a real hallmark, and it can show up quite early, sometimes even before clear symptoms appear in people known to carry the gene. That's important. Now, it's also really important to know about the negative markers for FFI because they help rule out other things. For instance, unlike many forms of CJD, spinal fluid tests in FFI are usually normal. That 1433 protein you often see elevated in CJD, typically absent in FFI.
Interesting.
Yeah, and the EEG, the brainwave test, usually just shows some nonspecific slowing. It doesn't typically have those periodic sharp wave complexes that are characteristic of most CJD cases. And a standard MRI scan of the brain often looks surprisingly normal, especially early on, unlike CJD, which can show specific bright spots in certain areas.
So finding nothing on some tests is actually a clue towards FFI sometimes.
Exactly. It helps narrow it down. When distinguishing it, the main things to consider are sporadic fatal insomnia, SFI, looks almost identical but no mutation, and that other form of familial CJD linked to the same mutation but with valine at 129, D178N 120V. That usually presents differently, more cortical dementia. So FFI and SFI share that PET scan thalamic shutdown and the sleep study chaos. CJD often has the positive EEG-CS fMRI findings and more cortical signs. It's a puzzle.
It really sounds like a precise diagnostic puzzle.
Okay.
With such a devastating prognosis, this relentless progression,
Yeah.
what does the treatment landscape look like? Is there any hope?
Yeah.
Anything at all?
Well, this is the toughest part. Currently, there's no cure. There's no treatment that modifies the disease course. Management is purely symptomatic and palliative. It's about trying to maintain comfort and quality of life as best as possible. And this is a critical point we need to emphasize. Conventional sleeping pills, like benzodiazepines, they are not effective.
Right.
In fact, they can often make the confusion and delirium worse. You simply cannot force sleep when the brain's fundamental machinery for sleep has been destroyed by the neurodegeneration. The insomnia isn't the root problem, it's a symptom of the brain damage.
That makes sense. You can't fix a broken engine by just pushing the gas pedal harder.
Exactly. That's a good analogy. However, the fact that FFI is caused by a single gene mutation, that it's monogenic, does make it a prime target for innovative molecular therapies. And the big shift in thinking now is towards pre-symptomatic prevention, because by the time symptoms appear, significant, irreversible neuronal loss has already happened.
So trying to stop it before it even starts.
That's the goal. There's actually a pioneering preventive clinical trial ongoing in Italy right now. They're giving asymptomatic people who carry the FFI mutation daily oral doxycycline, an antibiotic actually, over 10 years, to see if it can delay or even prevent the disease onset. Early days, but hopeful.
Wow.
But perhaps the most promising long-term strategies involve gene-targeting therapies. The idea here is to reduce the amount of the normal prion protein, PRPC, that's available in the brain. If you lower the amount of the raw material, the substrate, you can theoretically halt or significantly slow down that misfolding chain reaction.
Cut off the supply chain for the bad protein.
Precisely. This includes approaches like antisense oligonucleotides, ASOs. These are short synthetic strands of nucleic acid that basically bind to the messenger RNA from the PRNP gene and stop the protein from being made. They've shown success in animal models of prion disease. Looking even further ahead, you have gene therapy or gene editing possibilities, using things like RNA interference, RNAi, to directly silence the PRNP gene, or even more advanced techniques like CRISPR-based gene editing systems to actually go in and correct the D178N mutation in the DNA itself. There was recent research using a technique called base editing in a mouse model of prion disease that extended their lifespan by over 50%. Still early, but very exciting.
If we connect that to the bigger picture, these gene-targeting strategies, they really feel like the ultimate goal of precision medicine for FFI, and maybe for other genetic brain diseases too. It injects some real hope into what seemed like such a dark picture.
Absolutely. These approaches are aimed squarely at the root cause, and that's where the greatest hope truly lies for the future.
Beyond the intricate science, though, this disease clearly has such a profound human dimension. What is the real impact on patients and their families as they navigate this devastating journey?
Oh, the human impact is immense. It's truly devastating, not just for the patient, but for the entire family system. For the patient, as we've discussed, it's this terrifying, relentless decline into agitated wakefulness, hallucinations, delirium, losing physical control, losing cognitive function. And what makes it particularly agonizing is that patients are often acutely aware of their deterioration, especially in the earlier stages. That leads to severe anxiety, understandable depression as they face this.
Oh, wow, that awareness must be incredibly difficult.
It's hard to imagine, and the family's burden is, well, it's immense,
Yeah.
and it carries that unique genetic shadow. Caregiver strain is extreme. The rapid progression demands constant, often 247 care. This leads to emotional and physical exhaustion, burnout, and this profound sense of anticipatory grief.
Knowing what's coming.
Exactly. But maybe even more unique to FFI and similar genetic diseases is the genetic anxiety and existential uncertainty. First-degree relatives, children, siblings live with that stark 50% chance of inheriting this fatal condition. This forces incredibly difficult, life-altering decisions about genetic testing. Should I know? What do I do if I know? It profoundly impacts mental health, career choices, financial planning, family planning, whether to even risk passing it on. It casts this multi-generational legacy of fear and anxiety.
That's a heavy burden to carry.
It truly is. And then there are the practical aspects. The rarity often means a long, frustrating diagnostic odyssey just getting answers. Caregivers frequently have to cut back work or leave jobs entirely because the demands are so constant, leading to significant financial strain on top of everything else. For these families, good support systems are just absolutely essential. Psychosocial therapy, counseling is critical, and patient advocacy groups like the CJD Foundation are indispensable. They provide resources, helplines, connect families so they don't feel so isolated, and they tirelessly advocate for more research. They are vital.
This deep dive into fatal familial insomnia, it really underscores that intricate dance between our genes, our brains, and something as fundamental as sleep. It's a sobering reminder of just how complex neurodegeneration is, but also, I have to say, a powerful testament to the detective work of science in unraveling these mysteries.
Indeed. And despite these incredible strides, especially in understanding the genetics and the protein mechanisms, critical gaps definitely remain. Perhaps the single biggest unanswered question is still, why? Why does that specific D178N129M prion strain target the thalamic nuclei with such brutal selectivity? If we could unlock that mystery, understand the basis of that selective vulnerability, that could provide fundamental insights relevant to many other neurodegenerative diseases where specific brain regions are targeted.
Yeah, that could be huge.
Absolutely. Future research also urgently needs to focus on developing reliable biomarkers that can detect the disease process before symptoms start. Early detection is key for any preventative therapy to work, and ultimately, as we discussed, the most hopeful path forward really lies in advancing those gene-targeting therapies, ASOs, RNAi, gene editing, aiming to reduce or eliminate the PRNP gene expression itself, trying to stop the disease before it ever truly begins.
So wrapping this up, what does this all mean for you listening to this? Well, while FFI itself is incredibly rare, studying it really illuminates the astonishing precision with which a single genetic typo can wreak such havoc. But it also highlights the incredible potential of modern science to identify, understand, and maybe one day overcome even the most baffling and devastating diseases. And maybe consider this provocative thought as we finish. If one tiny switch, a single amino acid change guided by another common variation, can turn a normal protein into a fatal agent that literally steals sleep and life, what other subtle genetic variations, maybe interacting in complex ways, might hold the keys to understanding and eventually treating the much broader spectrum of neurodegenerative disorders like Alzheimer's or Parkinson's that still puzzle us so much today. It really makes you wonder how many other hidden molecular switches, like the one in FFI, might be out there, just waiting for us to find them and figure them out. Thank you for joining us on this deep dive into fatal familial insomnia.
We hope this provided you with some valuable insights into this unique and challenging condition.
Concierge Care, providing world-class, personalized care for patients and members. Taking a proactive approach by offering its free digital health clinic for anyone in need of these services. Reach out to us via our website, www.health.conciergecare.net, or give us a phone call at 786-809-2345.